And now if we compare with the disease model, and we are looking into periodontitis or gingivitis, which is one of the most common oral mucosal diseases, where you have an inflammation of the gum, and if it becomes very severe, what happens is that if this is your gum, and here you have the oral facing side of the gum and the tooth facing side of the gum, if you have a lot of inflammation in this tissue, with severe periodontitis, there is a retraction of the gum and you can have tooth loss. All right, and so what we have done, combining single cell with our spatial proteomics and transcriptomics, and here we're using Murphy's just with the 300 plex. This was a few years ago, and we can improve this, of course, for future studies. But what we could do was really comparing the different subpopulations of epithelial cells and fibroblasts within this kind of quite heterogeneous structures, where you have the tooth-facing epithelium. There is the one where you have accumulation of bacteria and plaque. And you could see these subpopulations here in orange of epithelial cells. They are expressing a lot of inflammatory cues. And so if we now combine these spatial coordinates of the cells with their gene expression on the MERScope data, we can really identify not only what are the cells, but actually the pathways that are upregulated in these perio diseases. And by looking at the signaling pathways, then we can look at the targets that are upregulated. Here you can see the different receptor ligand motifs that are upregulated in this region of inflammation. And then by running drug to cell, you can look at all the drugs that could eventually target these receptor ligands. So it's really a cool tool to allow you to then look for drugs that you can implement into new therapies. But most important for the Human Cell Atlas is also the possibility to, our vision to make the data available for all. And so all these data sets are deposited in Cell by Gene, which is a platform, and you can scan the QR code, which is a platform that allows you to look at the different subpopulations, select the tissue that you want specifically, select the different studies, so you still have the link for all the different studies so that you can credit every authors of the different studies. And then you can run differential gene expression analysis between the different subgroups and really start creating new hypotheses with these data sets. And the next questions on the Human Cell Atlas is really to compare the different developmental stages and how are these deregulated in disease. So we are already now compiling the different studies for disease studies for the oral mucosa, and then of course doing in vitro and in vivo validation of our findings. And from what I understand, I still have a few minutes, which is great because I'm going to finish by presenting one of the projects that is currently under revision and that started in skin, went to the oral mucosa, and this led us unexpectedly to the thymus. So I've been working in keratins throughout my career, particularly working on hair follicle, right? And these are intermediate filaments that initially were thought to be mostly cytoskeleton proteins, but there are more and more studies really highlighting that they actually may be playing a role also in immunity. And I got interested in keratin 76, which is expressed in the skin and in the tongue and buccal mucosa, mostly in the superbasal layers of the epithelial cells, so the most differentiated layers of the epithelial cells. And when I was during my postdoc in the lab of Fiona, we were able to see that we had a mutant for keratin 76, and my colleague Kiflia Katalin published a beautiful study really showing how the loss of keratin 76 affects the skin. When you have loss of keratin 76, you have hyperproliferation of the epithelial, the skin stem cells, giving rise to a thicker epidermis, and they also have an increase in inflammation. And I looked at these animals because there's an interesting phenotype also in the oral cancer, but let's leave that aside for now. But what we also seen is that there are some immune changes in these animals. The loss of keratin 7.6 leads to an increase in spleen and the lymph nodes, and there is an increase in the effector T cells and Tregs. And they also have an increase of immune infiltration in the skin, here with CD45. But if you look in detail, CD4 and CD8 T cells are increased in the skin. So how can keratins, like keratin 7.6, that can modulate the immune system and the T cells? For us, this was a really puzzling idea. And this work was started by Inês Tomás and Marina Torre back at King's, but then followed up by a very talented PhD student, now postdoc in my lab at Queen Mary, Mara Gelmetti. And Mara is an immunologist, and she really brought a new twist to this project that went from epithelial biology to immunology. We found that keratin 7.6 is actually expressed in some very rare superpopulation of thymic epithelial cells. And the thymus, for those that are not aware, is a mini organ that sits on the top of your heart and is important to create a functional T cell repertoire. So the immature T cells go through the thymus and are presented to tissue self-antigens and follow two phases of positive selection and negative selection. And if they are actually presented the tissue self-antigens and they have a quick affinity, they are competent and they go into the periphery. If they have a high affinity, if they recognize this antigen very strongly, they go through negative selection and die by apoptosis, and so that we don't have any kind of self-reactive T cells in the body. However, if by any chance we have a loss of a TSA, this T cell can miss this negative selection and they go into the periphery and they attack the self, creating an autoimmune disease. Until quite recently, we thought that air was this autoimmune regulator, was really instigating the expression of the TSAs, and they would have all the different thymic epithelial cells, we have a stochastic expression of these TSAs. However, recent studies in the lab of Diane Matisse have shown that this, and also in the lab of Kobe and in Israel, they have shown that this superpopulation, there are different superpopulation of thymic epithelial cells. Each of them are specialized to the expression of different TSAs. They don't all express all the TSAs, but there are superpopulations that are mimicking the muscle or mimicking or mimicking the microfold cells, and there's a superpopulation of thymic epithelial cells that are corneotecs that mimic the skin. So these are called the mimetic MTECs. And interestingly, keratin 7.6 is expressed particularly in the corneotecs, in the subpopulation of thymic epithelial cells that express skin-specific TSAs. And keratin 7.6 is actually expressing this structure called the Assel's bodies, where you have differentiated thymic epithelial cells. And for me, being a skin biologist was really interesting because they really follow the same differentiation pattern as the skin epithelial cells, right? Well, you have the keratin 14 in the basal, undifferentiated, and then you'll have more differentiated thymic epithelial cells that follow a similar program as differentiated epithelial cells in the skin. But we had a mutant for keratin 7.6 that had keratin 7.6 expression in the skin, in the tongue, in the buccal tissues, as well as in the thymus. So we needed to decouple the expression of this to the thymus only. So what we have done was taking thymic from embryonic mice, knockout mice, and graft these thymi into the kidney capsule of nude mice that don't have a thymus, and see whether you could mimic the loss of expression of keratin 7.6 only in the thymus and not in the other tissues. And indeed, we were able to show that just by removing keratin 7.6 in the thymus, we could have an increase of the spleen size and lymph nodes, an increase of the effector T cells, and an increase of inflammation of the skin without any problems in barrier defect or changes in the skin thickness, as we've seen in the total knockout. So it is the loss of keratin 76, specifically in the thymus, that is driving this immune phenotype. So we then explored, using single-cell RNA sequencing, what is changing transcriptionally in these thymic epithelial cells. And what we have seen is that in the keratin 76 knockout thymic epithelial cells, there is a decrease or a downregulation of the expression of skin-specific TSAs, TSAs that are presenting, antigens that are present in the skin. They are not expressed in the thymic epithelial cells. And what is, and you can see that all these markers are expressed in the mouse skin as well as in the human skin. So by losing keratin 76, we lose the expression of corneo-specific TSAs. But what's the mechanism behind it? How can the loss of a keratin, as suprabasal keratin, be leading into this loss of expression? Well, it seems that there is a change in the proportion of the different MTECs. Well, you have more undifferentiated MTECs and less MTEX3, which are the MTECs that are the corneotecs. And if you look at their differentiation using pseudotime analysis on the single-cell RNA sequencing, we can see that in the knockout, there is a delay in the differentiation of the thymic epithelial cells. And this was then confirmed by immunofluorescence, where you see less keratin 10 positive, which are the differentiated thymic epithelial cells. So we have shown that keratin 76 is important for the normal differentiation program of the corneotecs. And what's happening here is that looks like by losing keratin 76, there is a reduced T cell negative selection. And so we could see by doing Caspase-3 stainings that there is less cells going through apoptosis. And so there is a lack of negative selection because the T cells are not presented the skin-specific TSAs. And so to conclude, that we have shown that keratin 76 expression is necessary for the differentiation of the corneotecs, the expression of skin-specific TSAs in the thymus, as well as to the establishment of this immune tolerance in the periphery. So mechanistically, what's happening is that the keratin 76 MTECs specifically express skin TSAs, and then when the T cells bind to have a high affinity to those, they die or they go into the periphery, or if they don't, they have a weak affinity, they go into the periphery to form a competent T cell repertoire. However, when you lose keratin 76, the TSAs for the skin are not expressed. The self-reactive T cells lacks the negative selection shown by the the decrease of Caspase of apoptosis in the thymus. And then these autoreactive T cells go into the periphery and attack specifically the skin. So this is the first study that really directly links keratin 76 or any keratin with T cell tolerance, really presenting a novel role for the expression of keratins in the thymus and their importance in the development of skin immunopathologies. So as the take-home message from these 3-in-1 projects that have been developed in my lab and throughout my career as well, we started by looking at the skin and that led us into the oral mucosa, really understanding how the oral mucosa works to support skin regeneration. And that led us into thymus biology unexpectedly. But ultimately, what was more interesting is that this study on thymus and keratin 76 in the thymus really led us back to skin and to oral mucosa. So these tissues are both very diverse and much more similar than we imagined. These 3 projects really show how every tissue is a mosaic of unexpected cellular diversity. However, the biological mechanisms behind it are really creating surprising connections across the different tissues. And just to show that indeed these multi-omics technologies were essential for us to redefine both the boundaries as well as the bridges in tissue biology. And with that, I'd like to finish thanking all the collaborators that were involved in these projects, the team of the Human Cell Atlas, the funding that have supported these different projects, and the really exciting team of scientists that work with me, and they've been really fun to work with. And as the lab is moving into its new home, I would like to let you know that we have positions opening, both for MSc and PhD students, as well as postdocs. There will be a PhD program opening at the NIMSB with a call in the 1st of May for all the teams that are moving into the NIMSB. So stay tuned and connect with the new team at the NIMSB. And thank you so much for your attention.
Thank you very much, Inês. This was a really amazing journey through 3 different tissues. So thank you very much for this lecture. And please, if anyone has a question, you can raise your arm or just turn on the sound and ask the question.
So if I can ask first question. So Inês, amazing talk, very data rich, which I always like very much. I was wondering, on your oral data sets where you compared neonatal and adult, if I understood correctly, is based solely on single cell, but I was wondering if you saw any differences on other cells that the fibroblasts could interact with, or that interact with, that could also explain these different proportions or could build on those different proportions.
Definitely. So Diana has explored also the different proportions of the immune cells, for instance, and she's seen, for example, that macrophages are enriched in the skin, and she was also, and this is still work in progress, where we are looking into how the different subpopulations of fibroblasts also communicate with epithelial cells, but then also with the immune cells and the different programs between them. So definitely the immune cells. And then in terms of the epithelial cells, yes, there are differences, and the programs of expression, particularly on the suprabasal epithelial cells, are different between the oral mucosa and the skin as well. So we've seen differences there as well. And then there's another interesting study, particularly now when we are moving also into diseases like epidermolysis bullosa, and this is the work that has been done by Diana Pereira, as well as a master student in the lab that hopefully will become a PhD student soon, Alexandre. They have been looking into how these differences in vasculature as well. And I think all the spatial analysis that we've been doing with the phenocycler, with those samples, will really allow us to also look into the position of the different fibroblast subpopulations and the immune cells and the vasculature in these different tissues of healthy skin and oral mucosa versus chronic wounds. In the EB model. So yes, there are other subpopulations that we are exploring, and if you have anything in particular that you'd like to explore, we're always happy to collaborate and have a look at the data for your particular questions.
Okay, thank you.
Any other question from the audience?
I can ask a question. Hi, Inês. Nice talk. So if you could spend a bit more time explaining like part 1 of your talk about where the stem cells were dividing, because it remembered me the NMP, so the progenitors that make the spinal cord and the paraxial mesoderm. And I think my question is the old question of what makes a stem cell divide and so self-renew, and what makes them to differentiate. So if I understood correctly, so you have this pool of stem cells in this region, and there are different signalings depending on the position of these cells. You showed some sonic hedgehog as well. So and then you had a scheme of where you had these cells that they self-renew and that they make a specific cell type, and then a bit later they would make a different one. And so you suggested that this would be their plasticity in terms of potential. However, despite the fact that they become more transcriptionally committed. So I think my question is, how does this stem cell region changes over time? So the cell gives rise to another cell fate because with over time it ends up in a different region where you have different signalings, and also like all the stem cells in these different regions, do they self-renew, or do you have like pools of like long-term progenitor cells? I think, yeah.
Thank you, Andre. A question from a development biologist, and this is really what I love to discuss as well. So how does that work mechanistically? So you have the stem cell that only functions during anagen. This is not a long-term stem cell. This is an anagen stem cell that then disappears when the hair regresses, okay? And so what happens is that you have, can you see my arrow?
Yeah.
That you have the progenitor cells that is just opposed to dermapapilla. You have here a signaling of BMPs and noggin. So you have noggin in the dermapapilla and BMP4. Outside you have sonic hedgehog that is asymmetric, and this is something that no one ever described. There is an asymmetry on the sonic hedgehog, and it follows the angle of the hair follicle with the epidermis, with LGR5 on the other side. And if you know a bit more about it, I'll be very keen to discuss this further, because with Maria Kasper, it has been something that we've been discussed for several years, but we haven't published anything about it yet, because it's quite of a really niche, where there's this very small subpopulation here that expresses sonic hedgehog that is called the lateral disc. So this stem cell here divides asymmetrically. You'll have the mother cell staying connected to the dermapapilla, and then the daughter cell, so the transgenic amplifying cell here, number 2, we divide symmetrically to give rise to 3 and 4. These 3 and 4 then they start differentiating and they go into their layer. There are 3 or 4 progenitors for each layer here, and so they intercalate. So you have 2 doublets of one progenitor, of one transgenic amplifying cell, followed by doublets of another one that was not labeled. And we tried to do this with the confetti as well at some stage, but it was not clear to see all this organization because of some of the markers are nuclear in the confetti, so you lose the shape of the cell, then you don't know who is who, basically, in terms of the lineage. So then you have stem cell, transgenic amplifying, and differentiated cells that then start migrating upwards as the hair grows to give rise to the differentiated layers. Now the long term, those live in the bulge, which is in the upper region, in the bulge and in the hair germ. And we have another story. I'm happy to have a chat with you separately about it. Really looking at the contribution of the hair germ cells and the lower bulge cells to the renewal of the hair follicle, and how they are already somehow spatially patterned to give rise either to the cells that are surrounding dermapapilla or to the cells that will be the progenitors of the outer root sheet. And this is already kind of mapped from early telogen. And beautiful studies also from the lab of Valentina Greco have shown some pre-patterning of these long-term stem cells. Now, how this relates with the elongation of the embryo axis, this is something that is really interesting. You also see the intercalation of these cells that as you would see in the embryo, and then all the cells will be proliferating. The rate of proliferation is slightly different, and we've seen that in the human as well. And this is something that with more movies we can really study these dynamics.
Okay, and then this bulge is where you see the LGR5 expression.
The hair germ and the lower bulge.
Okay.
Which is also in the some part of the sheet and in the lower proximal cup in the lower part of the hair follicle.
Okay. And have you seen like differences in notch? Do you see some differences in
I have not. I think there's a couple of studies on that. I have not looked into notch.
because we are finding some interesting things regarding wind and notching in the NMPs, and that is this interplay that decides whether they will go for one fate or the other. Okay. No, very interesting, very interesting. Thanks.
Thanks, Andrea.
Hi, Ine. I actually have a question. So, thank you for the very nice webinar. I was thinking, what's very interesting in the difference in the fibroblast that you have in the newborn and then in the old mouse compared to the skin and the oral mucosa. And then I was thinking, could it be related to the resident microorganism that you have there? Because when you are newborn, the both mucosas, right, they're kind of cell. And then they will develop a microorganism that will be different for sure because it's a different environment. Mm-hmm. And then whether those microorganisms might affect, right, this distribution in the fibroblast could be something interesting. So I don't know if you're looking into it or you plan to
microRNAs?
Microorganism, like the
microbiome, yes.
Microbiome, right? So yes, I think that might be interesting because it's quite different, right, from the newborn and adults. And then it might have an effect on this what you're saying.
We have looked at, in a collaboration with Kevin Bird on the Human Cell Atlas, we have a paper on gingiva, and we looked at different microbiome and using RNA fish on the top of phenolcycler to be able to look at the different subpopulations of bacteria on the gingiva. And you can see the accumulation of those on the more inflamed side, which is really cool. Now, comparing skin and the oral mucosa, we haven't done it yet for the EB Atlas because of the chronic wounds in the skin versus the oral mucosa and the cancer incidence that is different. We collected swabs, but we haven't kind of run the metagenomics yet, but it's really something that we should explore. And from a single cell data, we can get this information. We just made an expert in microbiome to join the lab and really focus on that, which, there's only so much you can do, but this is something that we'd love to explore. And we have the data sets, so that we have matched single cell spatial and microbial swabs. But they are still sitting in the -80 for now.
Oh, that's very interesting because I also think maybe the spatial would be interesting in this, right? How those microorganisms are actually organized. And if you find any difference, maybe the good ones are in the mucosa, and can we use those in the skin sometime to help for the regeneration, right? So this is very interesting. That Phil, thank you, very nice.
Thank you, Javier. Thanks for joining today as well.
Any other questions? If we don't have any questions, I would like to thank Dinesh once again for a very interesting talk, and thank all the people that were present today. And keep watching this on our website. We will announce next names winner very soon. Thank you once again, and have a good day.
Thank you. Bye, Natasha.
Bye.
Okay, ciao Marta, thank you so much.
Thank you. Bye-bye.
Bye.